All right, hey, good morning. We're going to be looking at this lab today. We're going to be looking at how photosynthesis is affected by sunlight. So we're going to be using some bromethymol blue and Elodea plants to determine the rate of photosynthesis. So a couple safety things first. Your desk. You need to clear that off. So, second of all, make sure that your hair is put back. If your hair is like mine, you just don't want to get ruined. Okay. Then we need safety goggles. All right, so on your desk, really all you need is your textbook. You need it open to pages 30 and 31. So first thing we're going to do in a science lab is we're going to take our time. We're going to read the directions so that we know exactly what we are supposed to do today. So as you read over the steps, step 2, there's going to be a few things different. Mrs. DeBold and I, we've already mixed the bromethymol blue solution for you. So we already have that mixed. And what you are going to need to do with that, from these, you're going to get a smaller beaker. From the larger beaker, you're going to pour 100 milliliters. So look for the line coming from the bottom where it gets to 100. Pour it nice and gentle until it gets to about the 100 mark. There you go. You got enough bromethymol blue for you. All right, you're going to need only 2 test tubes on this one. The book says 3, but as a class, we're going to do just 1 control test tube per class. So each group is going to have 2 test tubes to actually test the independent variable, which we'll discuss, and I bet you can figure out which one that is. So next thing, bromethymol blue is an indicator. As you can see now, it is blue, but when we introduce carbon dioxide into it, it will turn yellow. So in order to do that, you're going to need to go get a straw. So you'll have to designate one person in your group that is going to exhale through the straw into the solution. Now you're doing this, please be gentle. Do not blow forcefully. Blow very gently. And when you need to inhale, remove your mouth from the straw. Breathe to the side. Do not inhale through the straw. So take it very gently, and you need to start blowing in there. Now you can notice on mine, if you can see, difficult to see on the camera, but now it's turned from blue to green. So now you need to keep blowing into it until it turns to yellow. So it might take another minute or 2. All right. As you see, it's difficult to see on the camera, but mine is very dark shade of yellow. I'm going to blow into it, exhale into it just a little bit more. All right, that should be good. Now the straw, you're going to need to dispose of properly, please. All right, so now you got your 2 test tubes. So make sure you remove the caps from them. Now you're not going to be able to have the stand at your table, so you're going to need to be extremely careful holding these test tubes. So now as you take them, pour it about halfway fill them up about halfway. And I can see each of them is about halfway. So at this point, you're going to need to see your teacher, do the mirror visit bulbs, and you need to get a couple sprigs of Elodea plants. So you're going to need to get 2 of them. We'll probably give you one chunk like this, and then you just need to break it in half, place it down into that. This is where you may want to use your straw. Just make sure it's pushed all the way down in there. At which point then you are going to take your bromomethanol blue solution and fill it back up till it is about 3/4 of the way to the top. Make sure your Elodea plant is completely submerged. As such, you're going to replace your rubber stoppers so that no oxygen or anything is getting inside. You're going to take a small sheet of foil, then you're going to take one of your test tubes, and you're going to completely wrap it in the foil. So this way, absolutely no sunlight can get inside of it. So now this is what we're testing. So you're going to make your hypothesis, which one is going to generate more oxygen over the next 24 hours. So at this point, we are ready to answer like step 6, where you describe the importance of having a tube that does not contain Elodea. That is your control. As a class, we'll just have one control bottle for you. So now at this time, you can take all your stuff, put it back in the proper location, and clean up and resume working on your hyperdoc. So thank you. Keep doing science.